welcome to Bloodwork. Today I'm going to be talking about a study that was recently published about Burkitt's lymphoma. But this is actually really an opportunity to talk about, I guess, a social issue that affects scientific research, and that's why I'm sat here in front of this big world map in my research building. This was a EDI initiative to show the breadth of people that we've got working in our institute from all over the world. But even so, even though we've got this great representation, a lot of science is still very European and American biased. A lot of the way we do research is looking at these specific populations. Now, Burkitt's lymphoma isn't very common in European or American countries. Where it is common is equatorial Africa, mainly East Africa, areas where malaria is endemic. Now, we know that certain ethnic groups, countries, however you want to think about it, are going to be more predisposed to certain diseases. The most common one that you might think about is sickle cell trait, which developed in response to malaria. So it is, of course, more common in areas where malaria is endemic, or people who are from ancestries where malaria was endemic. And it means that you need to actually consider this when you're doing research on these types of diseases. Now, the study that I've been looking at, they were looking for genetic variants, so bases in the DNA, which are linked to likelihood of getting Burkitt's lymphoma. So these are not mutations that are actually causing the disease, but things that might predispose you to be more likely to actually get Burkitt's lymphoma in the background of having something like Epstein-Barr virus or malaria infection, which is really what kickstarts the process. Now, looking at your genetic variant, a lot of the way we do research is by comparing the DNA to what's called a reference genome. This reference genome is made up of some anonymous donors' DNA that has been sequenced over many years, but it is 93% from one guy who has mainly European, some African ancestry, and then there's about 7% of it, I think, is from other backgrounds. But it is mainly this one guy. So this isn't necessarily the strongest way to look for specific changes in the DNA that might be linked to certain ancestral traits. So in this study, they took DNA samples from a whole load of children with and without Burkitt's lymphoma. They took them from Kenya, Uganda, Tanzania, and Malawi. So these are areas where Burkitt's lymphoma is common, where malaria is endemic, where they should have the ancestral DNA that actually matches to predisposition or not of this disease. So they're comparing these directly, and they did what's called a genome-wide association study. This basically just looks to see if certain regions of the DNA are more linked to the disease. With this approach, they found an area of the genome which was linked to the disease, and they found certain what are called single nucleotide polymorphisms, which basically just means an A is more often a T in some patients. They found these 17 of them in one locus, and this locus they'd found what it was, and they linked it back to a gene called RUNX1. Now, RUNX1 is actually the gene that I work on as well. It's a really important gene in blood development. It is often mutated in blood cancers. That's not the case here, though. These mutations are not in the coding region of the gene. They're not actually changing the protein that is produced for RUNX1, but they're changing a little bit how it's regulated. And this is quite cool. Because it means that certain variants, if you have them, mean that even in the background of having a malaria infection, for example, you may be protected from Burkitt's lymphoma. or you may be protected from dying of Burkitt's lymphoma, more importantly, because actually this is the most common childhood cancer in these areas. It has a really high mortality rate in these areas due to the healthcare system that is available. So these variants are altering how the cells are responding to the Epstein-Barr virus or the malaria parasite. They're potentially regulating a little bit how these cells are growing. So Burkitt's lymphoma starts in your B cells. It's a type of non-Hodgkin's lymphoma. So actually, if these cells are maybe a bit more relaxed when they've got this virus, they're not going to become cancerous. They might be able to repair their DNA a bit better and not drive this cancer. Now, at the moment, this is just an association study. They found something that is maybe underpinning the disease and how it develops. So at the moment, by itself, this isn't particularly helpful. But as global healthcare is improving, and this is really something that people are starting to acknowledge, that we need to not just fix cancer in rich European countries, but actually globally improve healthcare. This might be a way that children could be screened to see who's more likely to get Burkitt's lymphoma, who needs a vaccine against malaria, something like that. As the treatments improve, this is going to be a way to prioritise those who really need it, those who are going to die of this cancer otherwise. So, it's a start, but actually really thinking about the groups of people that you are studying and the background of their disease, the background of who is going to get it and who you are looking at is really important. And actually having representative cohorts is something that we are increasingly acknowledging in science. So having just wealthy, middle-class, white donors field samples, it's easier to engage them in England, we know this, but actually reaching out to these underserved populations is going to help improve outcomes for everyone, and that's really cool. So I will leave it there, and I will see you next time, hopefully with a special guest.